Hi, Gen Bio. Dr. Fry here. I hope you guys are all doing well and enjoying your Thanksgiving break. Today we are going to talk about population genetics, and this is the lab for which we handed out those orange and black beads. So you will use those as you work through your lab manual this week. Now, if you happened to go home a week early or a few days early and you didn't get the black and orange beads, you will need to find objects to substitute for the beads. So you basically need 180 items of two different colors, 90 of each. Okay, so you're gonna need either 90 beads of one color and 90 beads of another color, or you could use coins or even cut up different colored pieces of construction paper and use those. So that's what we are going to use to represent the frequencies of a dominant or a recessive allele in a population. Okay, so I'm going to give you the background here first. I'm going to do the PowerPoint, and then I'll be also posting some answers to the questions that are in the lab manual. Okay, so I'll post those and maybe even take pictures of my lab manual just to help you with a few additional notes. If you want to work through yours, then you can check your things against mine. That would be perfectly fine. All right, so first off though, again, we're talking about population genetics, and a few of the key terms that we have to get under our belt first are genotype, allele, and phenotype. Okay, so genotype is the specific allele makeup that a person has. So for instance, a person has two copies of a particular gene, one from mom, one from dad, right? And in the most simplistic view of genetics, there can be a dominant and a recessive allele, which are two different forms. So for example, again, this is probably oversimplified, but you could have a gene for eye color, and one allele could encode for brown eye color, and the other allele could encode for blue eye color. Okay, so those would be the alleles, the different forms, but the gene would be for eye color. And then the genotype is a person's specific makeup. So say I have one of each. Okay, I could have a dominant and a recessive. Then I would have what we call a heterozygote genotype. Okay, all right, so then phenotype is different because that is what we actually observe. We don't necessarily know, looking at a person, what their genotype is, but we can describe the trait that that person has. Okay, so for instance, you might see that I have brown eyes. That could mean that I have two copies of the brown, say, dominant allele, or I could have one copy of the dominant and one copy of the recessive. Okay, and you don't know which one my genotype is, but you can tell my phenotype because it's an observable trait. Okay, so in the picture here, what we're showing you is that there are three different individuals in all of the possibilities of this simple Mendelian genetics. There are three individuals who could possibly have the dominant trait, right, out of the total four. So there's four different siblings. Mom and dad have passed on either dominant or recessive traits. The options are that the children have two copies of the dominant gene. They could have one copy from mom that's recessive and one copy from dad that's dominant, or it could be the other way around. Dad could have been the recessive and mom could have been the dominant. Okay, so they have two chances of getting this big A, little a, this heterozygous genotype, or the child could have two copies of the recessive gene, so little a, and therefore that one person has a chance of demonstrating the recessive phenotype. Okay, all three of these will show that dominant phenotype, even though they could have two different genotypes. They could be a dominant, dominant, big A, or they could be dominant recessive or recessive dominant, okay, and have that big A, little a genotype. And like I said, the only option if they show the recessive trait is that they must have 2 copies of the recessive allele. So little a, okay. All right, so moving forward, we're talking this week a lot about the Hardy-Weinberg principle, okay? Hardy and Weinberg were individuals who theorized that allele frequencies within a population and genotype frequencies within a population are what we call in equilibrium throughout generation, from generation to generation, unless there's some disturbance. So what I mean by equilibrium is that the frequencies are not changing from generation to generation. Theoretically, the frequencies of the alleles and the frequencies of the genotypes should stay the same throughout time, unless there's some force that pushes one frequency out of proportion, or makes one more beneficial to the species, and therefore that one might be selected for. So some of these disturbances to equilibrium might include non-random mating, random mutations, selection, and what we call random genetic drift, which is just when we don't know what causes it, but there's some drift that results in some alleles having a higher frequency than others. So non-random mating and selection, these are both when there's a reason that one allele or one genotype is selected for within the population. Mutations can pop up sporadically and just be random, but any of these would be a disturbance, might cause the frequencies of alleles and genotypes to be changed from one generation to the next, okay? All that being said, in nature, there's frequently a disturbance in effect. There's frequently some factor that causes maybe one trait to be more favorable or more beneficial to the species than another trait. And so that might happen and shouldn't be too shocking when it does, okay? So the amount of change that is caused by these disturbances can then be compared to what we expect, the baseline, the non-disturbed frequency, okay? So when you compare what you see in a population to what you expect based on equilibrium, then you can hypothesize that there is some disturbance taking effect on the population, okay? Now, after a disturbance, this equilibrium should theoretically return after one generation of random mating, random mixing within the population, okay? All right, so Hardy and Weinberg put together 2 equations that sort of sum up our basic understanding of population genetics. So the variables P and Q represent the frequencies of the dominant and recessive alleles respectively, okay? So these are just variables. They can represent how frequent the dominant allele is in the population, so that'd be P, or how frequent the recessive allele is in the population, that's Q, okay? But when you have this simplistic, there's only 2 different versions, a dominant and a recessive form of the gene, when that's all you have, then that total P plus Q is going to make up all of your options for alleles for a specific gene. So that's going to be set equal to 1. And if the total is set equal to 1, you know that P and Q both have to be a decimal less than 1, okay? So P is going to be a decimal less than 1, Q is going to be a decimal less than 1. They could be equal. They could both be 0.5 if the dominant allele and the recessive allele are equally prevalent in a population, but they don't have to be. One could be more prevalent in the population than the other, and that would be represented if you had, say, P equal to 0.6 and Q equal to 0.4, but either way, they have to add up to 1, okay? Because that's going to represent the population as a whole. Okay, so those P and Q, as I said, represent the frequencies of the dominant and recessive alleles, respectively. Now, there's another equation here. P squared plus 2PQ plus Q squared is also equal to 1. So this is going to be the entire population, again, 1. And P squared, in this case, is going to be the frequency of the homozygous dominant individual. So any individual who got 2 copies of that dominant trait, that dominant allele, then that proportion of the population is represented by P squared, okay? Those individuals who are heterozygotes, so they have 1 dominant and 1 recessive, so either of these guys here, this is represented by 2PQ, then the proportion of the population, the frequency of individuals who have a heterozygous genotype. So you've got a big A, a little a, 1 copy of the dominant, 1 copy of the recessive, and those are the folks that make up the 2PQ variable, okay? And then the last option is that you have some individuals who have 2 copies of the recessive allele, okay? And that is represented by Q squared, okay? So these are all the people who are little a, both recessive copies. And so therefore these guys, you have to remember, are all going to display the recessive phenotype, right? Whereas P squared and 2PQ, these could all demonstrate the dominant phenotype, okay? We don't know, of those that have the dominant phenotype, we don't know if they're P squared or 2PQ individuals. But we do know that anyone who shows the recessive phenotype is going to be a Q squared person, okay? Person with a homozygous recessive genotype. All right, so let's move to our next slide. All right, so we have you working through, using beads, working through the lab manual, situations where the population has equal gene frequencies. So P and Q are equal in the population. That means they both equal 0.5. And we are going to have you go through an exercise where that is the case, okay? So equal prevalence of the dominant and the recessive allele within a population, okay? So P, the frequency of the dominant allele, which we sometimes call big A, is going to be 0.5. Q, the frequency of the recessive allele, little a, is going to equal 0.5. And again, they both have to equal 1, but we're okay, they do. All right, so this is, again, remember, this is the frequency of the allele, not the frequency of the genotype or the phenotype. Right now, we're just looking at alleles, not people, okay? Then once we take it a step further and we say, what would you expect the frequency of homozygous dominant individuals to be? We're talking about people now, or genotypes. Those that have the big A, homozygous dominant. They both 2 copies of that dominant allele. Well, using our second equation here, this one, P squared, we know we can find out based on the fact that we already know P, okay? So if P is equal to 0.5, then P squared is going to be equal to 0.25, okay? And the same process we follow through to find out the frequency of heterozygotes within the population. So just take 2 times P times Q. So 0.5 times 0.5 is going to be 0.25. And then times 2 is going to be 0.5. Okay, 2 times P times Q. Okay, gets you that half the population, 0.5 or 50% of the population, these guys are heterozygotes. Spelled that wrong. Okay, so then, and lastly, same concept. We want to know the frequency of homozygous recessive individuals, or Q squared. We can determine based on the fact that we know Q. 0.5 squared is going to be 0.25. Okay, so that's pretty simple. As long as you have either P or Q, either of those variables, then you can figure out all the rest. If you have one of them, you got the rest, because you know that P plus Q has to equal 1. And once you have those, you can determine the frequency of each of the genotypes and each of the individuals, okay? All right. That same concept follows through even when you have unequal gene frequencies within a population. So your alleles don't have to be equally proportioned within a population. They don't have to be 0.5 and 0.5. If you're given a situation where you are told that the frequency of P is 0.64 and the frequency of Q is 0.36, you can still determine the frequency of homozygous dominant individuals, heterozygous individuals, and homozygous recessive individuals just by squaring P, squaring Q, or taking 2 times P times Q. Okay. So same concept, but just make a note that the frequency of the alleles don't have to be equal within the population. Okay. Now, the third exercise that we're going to have you do, working through your lab manual, is a homozygous recessive lethal scenario. So this is a situation where any individual who inherits 2 copies of the recessive allele dies. So automatically kaput, gone from the population, okay? They do not survive. And this could happen through an important mutation in a gene that influences development in the embryonic stage, or other situations. Certain diseases do this. So assuming a situation where all of your homozygous recessive individuals die, they will not continue as generation 0, the original generation, changes to generation 1, the following generation. They will not be able to reproduce and contribute their genetic makeup to the following generation, okay? So you have to assume that the expected number of individuals with each generation is going to decrease, and that the proportion of alleles will also change as a result from one generation to the next, okay? So generation 0, this is our initial generation, could start with equal values for P and Q. Say they both start equal and they're 0.5. So 0.5 plus 0.5 equals 1. That makes up the whole population, okay? And from there, as we already demonstrated, we could determine the individuals, the proportion of individuals who are P squared, or homozygous dominant, 2PQ, or heterozygotes, and Q squared, are homozygous recessive individuals. Now, these poor fellows here, all these Q squared people, they're going to die. So they're not going to pass on that proportion of the alleles to the next generation, okay? So Q, the recessive allele, is the frequency of that is going to decrease with every generation as those individuals die, okay? So what we're going to have you do is we're going to have you work through this with beads. You're going to pull out your beads, and if you get, say, a black and an orange, that's obviously a heterozygote. If you pull out 2 black beads, that's going to be a homozygous dominant. And if you pull out 2 of the orange, that's going to be your homozygous recessive individual, okay? So you're going to do this, and the results that you get using the beads are going to be your observed results, okay? So you're going to keep track of that in your lab manual. But we're also going to ask you to look at what expected results you will see. So as I said previously, the Hardy-Weinberg principles assume, they expect, that the frequencies of alleles and genotypes will remain consistent from one generation to the next unless there's a disturbance, which clearly this situation is. The lethality situation is a pretty big disturbance on the population, on the frequencies of these alleles, okay? So you will use a formula here, an equation, to calculate the expected frequencies and the expected number of individuals that will be present in 5 different generations, considering the lethality of the homozygous recessive individuals, okay? So you're going to have to calculate this using this equation. Qn is equal to Q0 divided by 1 plus n times Q0. And before you get too confused, I'm going to break this down and tell you what each of those variables means, okay? What each of those letters mean. So n is just your generation number, whatever generation you're in. So like I said, generation, the first one is actually going to be generation 0, okay? And that we're starting with, we're going to say that P and Q are equal, they're both equal to 0.5. Okay, so we know that, and we know the proportion of individuals in that generation already, okay? But every subsequent generation, you got to calculate. So n is going to be the generation number. So if n is equal to 1, we're talking about the generation that is directly following generation 0, or our initial generation, okay? So that's just our generation number, 1, 2, 3, 4, okay? And so you will just plug those numbers, 1, 2, 3, and 4, in as n in this equation, okay? Now Qn is Q at generation, whatever you're at, whatever n is equal to. So if we're talking about generation 1, the one directly following generation 0, then we're talking about Q, the frequency of the recessive allele in generation 1, okay, after that first generation, okay? So Q1 is equal to the frequency of the recessive allele during the generation directly following the initial generation. All right, so that is what you're looking for. You don't know that, you want to find that. And you can find that by plugging in the other numbers, okay? So Q at generation 1 is going to be equal to Q at generation 0, which we already know. We know it's 0.5, okay? And then divide that by 1 plus the generation number, 1, 2, 3, or 4, times Q at generation 0, which again is 0.5. So really the only thing that you're changing from one generation to the next is going to be this n, but you're looking for Q at that generation, okay? You want to find Q, because once you know Q, then you've got P, because you know that Q plus P equals 1. So if you know what Q is in generation 1, say it's 0.4, then you know that P equals 0.6, right? So you can find that. And from there, again, you can find 2PQ, you can find Q squared and P squared. So once you have one of these variables at the generation, then you've got the rest of them. You can figure out the rest of them, okay? All right, so I have put together a little worksheet that if you would like to use, I'm gonna post it on Canvas. It looks similar to what I've put right here, but it just sort of gives you a space to do all these calculations for the 4 generations that we want you to look at. So once you have Q, by using this formula, you can get P by just doing 1 minus Q, and you can also get Q squared, P squared, and 2PQ. So you can just work through that. I just sort of talked you through how to do Q at generation 1, but you do the exact same thing. Q at generation 2 is going to be, just plug it in, Q at generation 2 is equal to Q at generation 0, which is 0.5, divided by 1 plus, this time we're changing the n to 2, okay? Multiply it by Q at generation 0, which is 0.5, and that'll give you 0.25, or it should. Okay, once you have that, you got P, and so forth and so on. So you're going to continue and do all those. Like I said, I have a worksheet. If you want to just do it in your lab manual, if you find some blank space, that's fine too, or scrap paper, whatever works. And from there, we can use these numbers to determine the number of individuals of each genotype at each generation, okay? So these are the frequencies of the alleles, P and Q, and the genotypes, Q squared, P squared, and 2PQ. Now they're not the numbers of people though. So if you start with a population of 100, you just multiply 100 by the frequency of whatever variable you're looking at to get the number of individuals. If you start with a population of 90 and you know that your P squared is 0.25, then you multiply 90 times 0.25, and you get 22.5, which since you can't have half people, you got to round up. So you've got 23 people that are homozygous dominant, big A, or P squared, when you started with 90 and you multiplied it by the frequency, P squared, okay? All right, and so forth. Same for the heterozygote people, the heterozygote individuals. You multiply the total number of people alive in that generation, we're starting with 90, by the frequency of 2PQ individuals, and you get a number of people then, okay? And the same thing, 90 people times Q squared at that generation gives you the number of people who are homozygous recessive, okay? So you can do the first one before anyone dies off, this first generation, generation 0, before anybody dies off, but then you can't do the second row here until you've actually observed results using your beads. You have to pull out your beads and find out how many people are alive to start that generation, okay? So anybody who was homozygous recessive, those people died and have to be taken out of the population. So you're going to have less people starting the next generation, okay? So I just gave you an example, but you're going to plug in the number of people alive based on your observed results using the beads, okay? And you multiply the number of people by the frequency of each of the variables that you're looking at, okay? All right, and so what this table gives you then is the expected number of individuals of each genotype at each generation, okay? And the reason we want you to find the expected number is that we're comparing the observed results with your beads to the expected results, which you determine mathematically, okay? And the reason we're doing this is we want you to compare them and see if that makes sense, if it's statistically sound, statistically what we would expect, okay? All right, so when you are doing some of the additional problems that are in the lab manual, there's some problems in the back, last couple pages. These are the basis, I believe, for the exam questions. So the problems that you do in the back of the lab manual are usually included as extra credit points on your lecture exam, okay, your final exam in lecture, okay? So when you're doing these problems, a few things to keep in mind. Just first thing first, figure out whether the question is asking you to work with alleles or with genotypes or with phenotypes, okay? If it's asking you to work with alleles, you're just talking about P and Q, okay? P plus Q equals 1, and it's simple. If you're talking about genotypes and phenotypes, now you're working with P squared, 2PQ, and Q squared, okay? And something else to keep in mind is that the dominant phenotype can be either P squared or 2PQ as far as the genetic makeup. Whereas the recessive phenotype individuals, they have to have that Q squared genotype, okay? Just keep that in mind. The question says all the recessive people equal this number and all the dominant people equal that number. Well, the dominant people could be in 2 different categories. You don't know. You do know that the recessive trait has to correspond to the Q squared individuals, okay? All right, and lastly, just try to pay attention to whether or not the question asks for observed or expected results, okay? So whichever one you're working with at the time, you're gonna be asked to observe the frequencies using your beads, pulling out sets of 2, those are your observed results. Then you're comparing your observed results with the expected results, which we determined mathematically on the previous slide, okay? And in order to compare those two, we're gonna use the chi-squared test, which I know we've used before during the course of the semester, but just as a rehash, chi-squared equals the sum of each of the observed values minus the expected values squared divided by the expected values, okay? So you're gonna use the observed as the number that you pull out with the beads, the expected as what we expect based on the Hardy-Weinberg calculations. And then don't forget to do all 3 and add them together to get your chi-squared value. So it's the sum, okay? So you're gonna do that observed minus expected squared divided by expected for each of these groups, the big A, homozygous dominant, the heterozygotes, and the homozygous recessive folks, okay? And then you add all those values together to get your chi-squared value. Once you have that chi-squared value, you want to turn to your chi-squared distribution table, which there's one on the next slide, and there's also one in your lab manual, okay? So you want to use the number for degrees of freedom equaling 1. Okay, so we have 2 alleles, dominant and recessive, and so we subtract 1, and so we usually use 1 degree of freedom for this, okay? And then if your chi-squared value, like we did before, is greater than the critical value, which we look at using a p of 0.05 and 1 degree of freedom, then it's probable that the difference between your observed and expected results did not arise by chance and therefore is significantly different, okay? So if we see a significant difference between the observed and the expected results, then this probably means that the phenotype is not in equilibrium, okay, for whatever reason. All right, so let's move to our chi-squared distribution table. So for us, again, we're using a p of 0.05 and 1 degree of freedom. So you're looking to see if you have met or exceeded the value of 3.841. If you have not, then you have to assume that everything is in equilibrium, okay? So as I said here, if we see any significant difference between the observed and the expected number of individuals, this means the phenotype is not in equilibrium. But if it's within like the normal range here, anything below p of 0.05, anything below 3.841, then it is in equilibrium, okay? All right, so now I put together a couple slides just walking you through 2 of the problems that are in the lab manual. So I want to do a couple with you, especially because I'm not actively in lab, to help you work through these questions in person. So this first problem is identical, or it should be, to the problem on page 125 in your lab manual, okay? So the problem, I'm gonna walk through it with you, okay? And we are going to play this so that hopefully I don't give you all the answers right away. I want you to actually work through with me. So take out your calculators, take out your lab manual, and we're gonna work through how to do these problems so that you can do the others on your own. There's a whole bunch of questions. We're just gonna do 2 together. I will also post the answers, but I'd like you to try to work through as many as you can on your own, okay? So let's do this first one together. Cystic fibrosis is a recessive trait that affects 1 in 2,500 people, okay? The first question is, what is the frequency of the recessive allele? Okay, so given this information, cystic fibrosis is recessive. It's a recessive trait, and it affects 1 in 2,500. Based on what I said before, I'm hoping that you're honing in on the fact that it's a recessive trait as a key to what this means in terms of P's and Q's, okay? So recessive trait, remember, has to have a genotype of Q squared, okay? So Q squared is going to equal those individuals that are represented by 1 in 2,500. So 1 divided by 2,500 will give you your variable of Q squared, okay? So if you do the math, you break out your calculator, 1 divided by 2,500 should get you 0.0004, okay? So that's Q squared. Now, once you have this, and you're pretty sure that you're correct on any one of these variables, Q squared, Q, P, whatever it be, you're home free. You can get the rest, okay? So once you have 1, Q squared P equals 0.0004, you can easily find what Q is by taking the square root of Q squared. So Q squared equals 0.004. The square root of that is going to be 0.02. Okay, just take the square root of it. Now, once you have q, you know that p plus q equals 1. So take 1 minus 0.02, and you get a p-value of 0.98. Okay. So now the next question in your lab manual for this one is, what is the frequency of the homozygous dominant individuals? So again, you have to think, what does this mean in terms of P's and Q's? Okay, frequency of homozygous dominant, that's going to be P squared. So to get P squared, you just have to square your P value. Okay, so if you get 0.98 times 0.98 or squared, you should get 0.9604. Okay. All right, and then the frequency of the homozygous recessive individuals, we actually already did that one. We know that from the beginning because that's what the question told us. The homozygous recessive individuals, this equals the Q squared variable, okay? So we know that one already, but if we didn't, we could just reference that it's Q 0.02 times itself squared, okay? So that's Q squared. Frequency of heterozygotes, you just take 2 times P times Q, and you ought to get 0.0392, okay? All right, so now we have the frequencies in a population, but we don't know what this means in terms of numbers of actual people because we didn't, up to this point, know the population size. Say you have a population of 75,000 people. All you have to do now to figure out the number of people for each of these genotypes is to multiply 75,000 by each of the different variables. So if you want to know homozygous dominant individuals, you're going to look at your P squared, which is 0.9604, and multiply that by 75,000. Okay, that's the number in the population. So what you should get then is that 72,030 people are homozygous dominant. Okay. All right. So next, I just had to continue on to the next slide, but yours continues directly below in the lab manual. Next question is, in a population of 75,000, how many are heterozygotes? Okay. So again, I just copied from the previous page. We already knew that the proportion of heterozygotes is 0.0392. That's 2PQ, okay? Now, if you multiply that by 75,000, you should get 2,940, okay? Same question, in a population of 75,000, how many this time are homozygous recessive? Well, we know that the homozygous recessive folks, the proportion of those are equal to Q squared, or 0.0004. So again, if you multiply 75,000 by 0.0004, you should get 30, okay? All right, so that is the first question. Next, I skipped to question number 3. So the ability to taste PTC, so you're gonna work through number 2 on your own, okay? I think it's similar to number 1. Number 3, we'll work through together, and then the rest you're gonna work through on your own, and like I said, I'll post the answers. And if you have any questions, you can always email me if you're not getting the correct answer and you don't know why, okay? All right, so number 3 is the ability to taste PTC depends on a completely dominant gene, big T, okay? So tasters can be either homozygous dominant, big T, or heterozygotes, big T, little t, okay? We don't know which one. They're one of the two. But non-tasters, we know have to be homozygous recessive or little t, okay? Based on what the problem is telling you. This PTC, which is a sour, bitter tastant chemical, can only be discerned, can only be tasted by those who have at least one copy of a particular gene, okay? So they have to have at least one big T, okay? If they have 2 little t's, too recessive, they can't taste it, okay? So the problem also tells you that a sample of 100 people showed that 84 were able to taste PTC. Okay, 84 of the 100 were tasters. Now, it asks you to calculate the gene frequencies first for the dominant and the recessive allele, big T and little t. Okay, from what we're given directly in the problem, we know that 84 out of 100, or 84 or 0.84, the proportion of the population, 0.84, these can be big T, or they can be big T, little t, but we don't know which. 84% of that population can taste it, but from there, we can't really determine the gene frequencies of big T and little t. What we need to know, what we have to remember, is that the remainder are non-tasters. Okay, so the ones that aren't mentioned, that group of people are non-tasters, and we do know their genetic makeup. Okay, so 84 out of 100, they can taste it. But the 16 out of 100, they can't, and we know that they have to be represented by that Q² variable. They're non-tasters, they're homozygous recessive, okay? So just keep that in mind. You don't know of that 84% if they're big T or big T, little t. They could be homozygous dominant or heterozygotes, okay? What we do know is that 16 and 100 are little t, two copies of the recessive, okay? All right, so therefore, what we can determine is that Q² is equal to 0.16. So 16 divided by 100 is 0.16. That's going to be equal to Q². And again, once we are pretty sure we've determined one of the variables correctly, we got all the rest, okay? Q², we're just going to take the square root of Q² to get Q, okay? So we know that Q then equals 0.4, okay? And because we know that P equals 0.6 because P plus Q have to equal 1, okay? So now we can go back and answer that first question. Calculate the gene frequencies for big T and little t. Well, big T is gonna equal 0.6, our P folks, and little t is gonna be equal to the Q folks, so, or alleles, that's gonna be 0.4, okay? Next question there in the lab manual asks you, how many heterozygotes should there be in a group of 100 people? Okay, so here we're talking about 2PQ, okay? And to find that out, all we have to do is multiply 2 times P, which is 0.6, times Q, which is 0.4, 2 times 0.6 times 0.4, and we should get 0.48, okay? So 48 people. Oh, I got too far ahead of myself. Okay, so how many people would you expect to be big T? Those are your P² people, okay? So P, which is 0.6, squared is going to be 0.36, and if we're talking about 100 people, it's 36 people, okay? All right, so that is the third problem. Like I said, work through the rest and see if you have any trouble. I will post some notes about those and additional notes showing you how I would work through the lab manual. I'll just take pictures of mine. And again, if you have any questions or you're not sure, this is kind of a tricky one to do from afar. So please reach out if you're having any confusion at all, okay? Just shoot me an email and I'll be happy to help you. All right, hope y'all are doing well and have a great Thanksgiving. Take care, and like I said, you can always email cfry@lvc.edu if you have any questions.